First, a big thank you for being here. I do wish that we could do it in person so this presentation could be more interactive, maybe next year. For now, as you listen, please jot down your questions and we will have plenty of time to address them. Today, I'll discuss the partnership between researchers, clinicians, and Fanconi individuals and their families. I will start by saying that I truly care about individuals with Fanconi anemia. I would not be doing the research that I'm doing if it were not for the struggles that you face. Yes, I do love to discover how our cells work. I love to have a deep understanding of how exactly the DNA, our genetic material, is repaired, and how cells without the Fanconi anemia proteins turn to become cancerous. But having interacted with FA families, watching the challenges that you face, the priorities for our studies have become much clearer. Understanding the biology of tumor development in Fanconi anemia so we can develop new treatments as well as prevention strategies is really driving my lab's work. So I'll give you a short bio so you know my background. I was born and raised in Poland in a family of scientists and physicians. I came to the States for college and eventually completed an MD PhD program. So I'm a physician scientist, and although I do not treat patients, I interact with patients as study participants. I trained as a clinical pathology physician, so I can interact with surgeons, pathologists, and other physicians very easily, and I can also interact with patients as they're enrolling in our studies. I got interested in how DNA is repaired during a PhD and started working on Fanconi anemia proteins in 2005 while being a postdoc at Harvard. And in 2009, I started my own laboratory at the Rockefeller University, and this is when I took over the International Fanconi Anemia Registry from Dr. Auerbach, who was running it since 1982 and was retiring in 2009. My laboratory, together with multiple collaborators, just completed a study of 55 tumors from Fanconi anemia individuals and describe the changes in the DNA that are present when the tumors develop. I'm on a scientific board of the Fanconi anemia Research Fund, and I'll be co-leading a collaborative team of 7 laboratories working on cancer and Fanconi anemia, and I also collaborate with other groups as well. And one of our goals is to bring better prevention and treatment of cancers and Fanconi anemia. So here's an outline of the talk for today. I will give you a broad introduction to Fanconi anemia. I will describe different registries as well as different Fanconi anemia cancer studies. I'll mention ongoing studies from the IFAR, the International Fanconi anemia Registry, but we will focus on human cancer for most of the talk. The information and tissue samples that are useful for research studies, and I'll also describe what we do with the tissue samples, talk about the process of getting information and samples, and mention some challenges and some solutions. So first, let's talk about Fanconi anemia as a disease. As you know, Fanconi anemia is a very complex disease. I think of it as a disease of stem cells. Stem cells are cells that can divide and make more of themselves, but they also can give rise to other cells that make a particular organ. So the fact that the stem cells don't function as well as Fanconi anemia as they should leads to a number of problems. These include developmental abnormalities of arms and hands, kidneys, and other organs, bone marrow failure resulting in the inability to produce enough blood throughout life, necessitating bone marrow transplant, as well as issue with fertility. The second feature of Fanconi anemia is predisposition to tumor development, including leukemia, tumor of the blood, as well as number of solid cancers, most prominently the head and neck cancers. Stem cell problems arise in Fanconi anemia because a very specific lesion in the DNA, our genetic material, is not repaired. We call this lesion a crosslink because it links the two strands of the DNA. And when the Fanconi anemia proteins are working, these crosslinks are fixed. But if Fanconi anemia proteins are not functioning, like in cells from individuals with Fanconi anemia, the lesions persist and then cause a problem. And you can visualize this problem as shown here. The crosslink is a boulder on the road, so the DNA cannot be copied, and that creates DNA damage that eventually leads to the problems we see in Fanconi anemia individuals. So the DNA damage that is not repaired shows up in the chromosomes. Normally, our chromosomes look like X's, which majority of the chromosomes shown here in this metaphase spread look normal. However, chromosomes which are broken can come together in structures that we call radials. These are shown here in blue. These are abnormal structures that we can elicit when we treat these cells with agents that make crosslinks. And this test can diagnose patients with Fanconi anemia, but this damage can also lead to cells dying or transforming into cancer. So understanding how this DNA damage is made is really important. So you may wonder where the DNA damage comes from. There are three sources. One is endogenous, coming from the cells and normal cellular functions. The second one is exogenous, like tobacco smoke, alcohol, or bacteria that live in our mouth. And finally, chemotherapeutics that are used in non-Fanconi anemia individuals for cancer treatment. These chemotherapeutics need to be avoided in Fanconi anemia individuals. And understanding of DNA damage and where it comes from is really important. One day we're hopeful that by boosting the detoxification systems in our cells, we will be able to do something about the endogenous sources of damage. However, one place where we can make a huge difference in Fanconi anemia and cancer development is by not smoking and not drinking. So Fanconi anemia patients should avoid smoking and alcohol at all costs because we know that in tobacco smoke and in alcohol there are agents that can create crosslinks that cannot be repaired by Fanconi anemia individuals. So that's sort of how we can help in prevention of cancer, for now at least. Okay, so this concludes this broad introduction to FA and DNA damage that occurs in Fanconi anemia cells. And now I'll move to discuss different registries that exist. One point that I want to make is that you can be in multiple registries, and as I'll discuss, there are multiple registries that exist, and until we unify some of them, your participation in multiple registries will be necessary. So the first registry that I'll talk about is the FARF registry, which collects demographic information and data from individuals themselves. The second one is the NIH registry, which was directed by Dr. Blanche Alter and will be continued by Dr. Sharon Savage and Dr. Neelam Giri. This is a very detailed, descriptive registry focused on epidemiology, which is incidence of certain features like developmental abnormalities or cancers. That registry has been very important for describing the disease. However, it has not worked with tumor samples. But we hope that this registry will be developed further and will be also combined with others and would serve especially patients who are at the age when there is a possibility of development of cancer. Third registry, which I'll describe in a moment, is the International Fanconi Registry, IFAR. And our IFAR registry is ideally set up to collect samples, and I'll describe the process. There are also many country-specific registries. I know there is one in Spain and France and in other countries. And it's through the collaboration with some of those registries we were able to get more samples for our studies of cancers. There are also other studies that are not part of really a registry. You are probably very familiar with the study of early diagnosis and brushings that was started by Ralph and Unique and is being continued by Unique and Christina. Dr. Pennington has a study concentrating on gynecologic cancers, vulvar and cervical, and collects information. And our registry, the IFAR registry, collects samples in parallel. So individuals who want to participate in Dr. Pennington's study will also be enrolled in IFAR. So for the rest of this talk, I'll concentrate on discussing International Fanconi Registry. This registry was started by Dr. Auerbach and Dr. Schroeder in 1982 to collect information and samples and start to understand the disease. It's actually remarkable that this is the time where when we didn't know any of the genes, and we did not even know with certainty that Fanconi was a disorder of DNA repair. Times have changed, and our registry has been involved in identification of new genes, and these are some of the genes that we've identified in the last 10 years. We're also interested in study of the mechanism of DNA repair. We study how a particular mutation shapes how the individuals with Fanconi present. So, for example, how early they develop bone marrow failure. We also want to understand how the Fanconi disease is modified. We want to explain why certain patients develop disease early or late. And finally, we also study patients with mosaicism, which is really important as it will help with understanding of potential of gene therapy. So now we will talk about goals of cancer research in general. The most important message that you should take from this talk is that research, although sometimes slow, can change your lives. It has done that for individuals with Fanconi in the area of bone marrow failure and bone marrow transplantation. With a lot of focus and funds, we as a field will change Fanconi as a disease, and that will for sure impact your lives. So your participation in research will improve the care of Fanconi patients and will eventually save lives. With that in mind, the goals of researchers working in Fanconi are to define how the tumors develop. We work on understanding of multiple tumors, including head and neck and anogenital cancer, but we also are starting to work on breast, liver, skin, and brain cancers. We want to identify the changes that are happening in the tumors that will give us clues about how to treat those tumors. And we want to identify the biomarkers for early detection. A biomarker is a substance or a change which is indicative of presence of tumor cells earlier than we can see, and that can potentially lead to faster diagnosis. Secondly, we are working to find best treatments for tumors. We need to gather information from previous treatments, what worked, what didn't work, and that's in order to inform future treatments. We also need to develop tumor models, including cells from tumors growing in tissue culture dishes. These are called cell lines, or growing while transplanted onto a mouse. Sometimes those we call patient-derived xenografts, or PDXs for short. We're also developing different mouse models. These tumor models can then be used to test possible treatments before we try them in patients. Finally, we need to concentrate on identifying and testing prevention strategies, and the mouse models that I described above will be really necessary for identification of how to prevent the cancer from developing in the first place. Eventually, we'll need to conduct patient clinical trials, and I believe you've heard about some of them earlier today. So next, I'd like to discuss differences between research and clinical care. The International Fanconi Registry is a research study. So we gather information and collect tissues to make new discoveries. Although I'm always happy to discuss what we are doing in the laboratory, no specific information is given directly back to the individuals or the family. If there is a relevant information that is revealed during research, we would communicate it with physicians, and the research findings then need to be confirmed through clinical testing. So, one thing to know is that research is slow. So, what we do will often not directly impact how the patient is being cared for. Physicians need to decide how to best care for the patient independent of research. So the studies for clinical care need to be done in a clinical laboratory independently of research. For patients who develop cancer, FARF has organized a tumor board, a meeting of number of specialists who discuss a specific patient and how to best care for that patient. And the tumor board will be really an important aspect of how we care for Fanconi individuals who develop cancer. So now let's discuss what our study collects. First of all, we collect medical information. How did the individual present? How old were they when they developed bone marrow failure? What kind of testing did they have? What kind of and how many hospitalizations, treatments, follow-up care? For those who had cancer, we collect surgical reports because that allows us to go back to the hospitals and get the tissue that was already collected. We're interested in reports from biopsies that turned out to be normal or just dysplastic on the way to become tumors. We also collect blood for individuals with Fanconi who are pre-transplant. For families without known mutations, we also collect parents and siblings. But for adult patients, we generally don't do that unless there's a particular question that we need answered. We also collect skin cells from which we grow fibroblasts, or skin samples from which we grow fibroblasts. This is true especially for adults who had a bone marrow transplant, because this is how we can grow the cells that were actually patient cells and not from the blood which was transplanted. And finally, we collect tumor tissue. So next, I'm going to discuss what we do with the samples that we collect. The blood that we get is separated into different components, and we collect white blood cells that include lymphocytes, and we perform breakage study on these cells, and we study those cells further. We also make a cell line, which is a collection of cells that come from a patient that can be propagated and studied in the laboratory. We can also share those cell lines with other researchers. As I mentioned, we also collect a small sample from the skin. It can be a biopsy of about 4 to 6 millimeter sample. It's the size of a pink eraser at the end of the pencil. From that biopsy, we grow fibroblasts, which are support cells from our skin. And fibroblasts are needed from patients who have had bone marrow transplantation. As I mentioned, the blood comes from the donor and not from the patient. And also, we like fibroblasts from patients who are mosaics because it allows us to study these cells further. Fibroblasts are primary cells that can be grown for a long time, and they're very easy to do experiments with. We can easily compare the DNA from fibroblasts to the DNA that is found in tumor cells. So now we come to the most important tissue that we collect, tissues from surgeries. The most valuable cancer tissue that we can get is a fresh sample. This is a sample that is collected by a surgeon and is kept in a special solution that preserves live cells. The cells are viable and can then be grown in the laboratory. We can grow these cells as organoids, which are collections of cells that look like tumor. We can also grow them in a mouse as a patient-derived xenograft, the PDX that I mentioned before. These are essential for testing therapies as well as for developing understanding of the environment of the tumor, which eventually may lead to new therapies as well. The second type of tumor that we obtain is the fresh frozen sample. This is a sample that was collected and immediately frozen on dry ice at very low temperature. This preserves the DNA of the sample, but unfortunately it kills the cells. This is a sample that can be used for sequencing of the whole genome of the tumor, which is very useful to understand what kind of genomic changes in the genome of the tumor occurred during the process of tumor development. And the final type of tissue that we collect is FFPE. This is formalin fixed and paraffin embedded tissue. This is a tissue that is put in formalin that preserves the structure of the tumor, but unfortunately makes the cells not viable again. These samples are great for sequencing of parts of the genome, not the whole genome, and those parts we call whole exome sequencing. They are very useful samples because they are archived by the pathology departments. So we can get these samples from past surgeries, even those from 10 years ago. And this is why we need to know if patients had previous surgeries and where they had them, because we can reach out to the pathology departments and ask for samples from a long time ago. So next, I'll discuss how we get information and samples. This is really where the partnership between the researchers, clinicians, and Fanconi individuals all come together. Somehow, I have to learn that there is a new cancer diagnosis from a physician, through FARF, through Facebook, or directly through communication with the individual. If the individual is already in IFAR, which is an ideal situation, I reach out to obtain releases so that I can talk to the treating physicians and obtain the records and tissue samples, those that were collected already, or those that will be collected in the future. If the individual is not in IFAR, they need to be consented before I communicate with the physicians. That can take about 30 minutes to an hour, and I go through the consent process, making sure that the individual understands everything about our study and has a chance to ask any questions. Participation in the registry, as in all registries, is voluntary and it is free. You can talk to me and then decide that this is not for you. Once the consent is complete, we can then start getting the information and samples from the physicians. And if there's a surgery and we know that there is a likelihood that there will be a tissue for research, I or someone from my lab will travel to the site of surgery. Or alternatively, in some circumstances, we will send a tube with special solution that then can be sent back by FedEx to our laboratory for processing. I want to stress again that any sample for clinical care are being collected first, and that's always a priority to collect whatever is needed for clinical care. Anything that's left over can be used for research, and that's what we strive to collect. So sometimes there's no tissue that's available for research, and in that case, we go back to the archived tissue that's been stored by the pathology department, tissue that was no longer necessary for clinical care. So my final slide is about challenges of participation in our research study and some solutions that we have. But I'm really looking forward to the discussion about these challenges and solutions in the Q&A session because I know that it's sometimes quite difficult to be in research studies. So the first challenge has to do with physicians. Even those who are associated with our studies often forget to let my team know about a biopsy or surgery. Some physicians taking care of Fanconi patients are amazing and want to help research. Some may not even be familiar with the necessity of research or are too busy to reach out. So, one of the solutions that we have is that the individuals with Fanconi anemia need to advocate for themselves, both for clinical care, which I know all of you do all the time, but also for research. And we as researchers and FARF as organization need to educate physicians about these issues, the need of samples as well as of information. The second challenge is that individuals with Fanconi anemia can be overwhelmed when things go into crisis mode, and certainly a diagnosis of cancer creates a crisis. It is not surprising that at that point clinical care is the most important to get right, and research becomes less important. So one of the solutions to this issue is enrollment into the registry before there is any crisis. That gives us a chance to enroll without time pressure and to gather all the information that is necessary for research before a crisis. If there's a surgery that after that, we can quickly communicate with the physicians and get samples if there are any. The third issue is that the process of getting medical records or tissue can be very long, as hospitals have different systems for storing and accessing information. So my team might have to get back to you a few times before we get all that we need. This definitely creates stress for the individuals with Fanconi anemia, and there is really not a great solution to this, except for us to be quite efficient and have all the signatures collected in one sitting, and maybe utilize Citizen as a platform, which we can discuss in Q&A. But overall, we really appreciate all your patience and assistance with all the studies. And if you do have PDFs, scans, pictures of results, please send them because that's one piece of information that we don't have to hunt from the hospital. And I'm really, really grateful for your participation. And if you don't know if you're enrolled, you can email me. We would obviously like all adults who are willing to help with this research to be in the registry, and that way we can have good longitudinal data for our studies. You can contact me at the email. Here's an office number, and you can also message me on Facebook which I use only for research studies. And this is it. I'll take any questions, and we'll hopefully have a nice discussion about participation in research. Thank you so much.